last two phytohormones, that is ethylene and abscisic acid. Ethylene is going to be the phytohormone which can be sometimes acting as promoter, sometimes act as inhibitor, but abscisic acid is a complete growth inhibitor. Okay, so let's start with ethylene. So ethylene is the only gaseous phytohormone. It's a simple hydrocarbon and a gaseous phytohormone, to remember that. Now, discovery of ethylene is very interesting. Here, Cousins discovered the ethylene by chance or by accident when he observed that the oranges, when they start to ripen, they were actually releasing some volatile substance, in the presence of which the unripe bananas kept nearby also had started to ripen up. Okay, so just see this. Cousins discovered and confirmed that ripe oranges would emit a volatile substance that brought about early ripening of unripe bananas kept nearby. Okay. Now, synthesis. Where exactly in the plant body will it be synthesized? It is synthesized in almost all parts of the plant body, but then mostly its concentration will be high in tissues which are undergoing senescence, or else fruits undergoing ripening. Okay. Precursor to this, to ethylene, is methionine, another amino acid. Now finally, moving to the functions. There are many functions of ethylene. After all, it's a promoter as well as inhibitor, so it has a long list of functions. Now, first is transverse and horizontal growth. As you know that gibberellin will be allowing lengthwise growth of the plant, but in comparison to that, ethylene will be rather allowing transverse growth of the seedlings, transverse or horizontal growth. It will inhibit lengthwise growth but will promote transverse or horizontal growth in seedlings. Senescence. Ethylene will be promoting senescence. It will allow aging of the plant parts, okay, especially the leaves. Abscission, that is shedding. Ethylene will also promote shedding of plant parts like leaves, fruits and flowers. Fruit ripening, very, very important part is fruit ripening. Now see, there are some fleshy fruits that have been observed to suddenly increase in the respiratory rate. In their respiratory rate, there will be a sudden increase prior to ripening. Such kind of fruits in which just prior to ripening the respiratory rate would suddenly increase are called climacteric fruits. And in climacteric fruits, it has been observed that ethylene will promote fruit ripening. Prior to the ripening process, when respiratory rate will increase, ethylene will be produced and will start getting released from the fruits, hence allowing ripening. Okay? And this phenomena of sudden increase in respiratory rate in such certain fruits is known as respiratory climactic. Okay? Now, breaking of seed dormancy. Now that you know that it is going to promote so many things, breaking of seed dormancy is one of the most important feature of ethylene as well. You know that seed dormancy, or seeds will be allowed to germinate under the effect of gibberellin. Similar to that, ethylene will also break seed dormancy or allow seed germination to occur. Flowering. In certain plants like pineapple as well as in mango, flowering process will be induced, or it will be fastened by the effect of ethylene. Sex expression. We have seen that in certain of plants like cucumber, ethylene will promote the femaleness in the plant body. You might be thinking, what is femaleness in the plant body, right? Femaleness simply means increase in female flowers. So in certain plants like cucumber, number of female flowers will be increased under the action of ethylene. And that's a benefit, because more the number of female flowers, more the number of fruits, isn't it? Next, elongation of stem in deep water rice plants. As you know, in rice, paddy fields, the rice seedlings will remain under the water. They will remain submerged in water. In such a condition, the rice seedling will be able to grow above the level of water due to the action of ethylene, which will promote rapid increase in the internodal growth and also of leaf base. Okay. So this is all about the functions of ethylene. Now moving to the usage, the most important usage of ethylene that is commercially very beneficial is fruit ripening. Now in fruits like mango, banana, etc., these particular fruits are kept, first of all, they are carried or transported to the godowns, where they are then allowed to, or they are sprayed with ethylene, and then that will promote fruit ripening. Now you might be thinking, how can we be spraying ethylene to a particular fruit when it is gaseous in nature? That's because ethylene is a gaseous volatile gas. That's why an alternative form of ethylene has been made in the lab that is going to induce ethylene formation, and that's an aqueous solution called ethipon. So it is ethipon which is sprayed onto the unripe fruits so that it is readily absorbed by the fruits, and slowly that will induce ethylene generation, or the fruits will then start releasing ethylene. As they start releasing ethylene, the fruits will start to ripen up. Okay, but of course, because it's not a natural process, the fruit ripening would not be naturally occurring here. It is induced. So in that case, the fruits might look very much ripened from outside, but inside they might not taste as good, right? Now, coming to the next one, that is abscission and thinning. In case of plants like cherry, walnut, etc., abscission of the plant parts like fruits or flowers, and thinning. Thinning means when excessive fruits grows in a particular plant body, that plant it gives out so much of its resource in formation of so many fruits that later on in the next season it might end up not giving any fruits at all. So hence thinning or shedding of those excess fruits from the plant body to reduce the load in the plant body is very essential, and that is going to be brought about in certain plants like cherry, walnut, etc. by the application of ethylene. Next, increase in number of fruits. Of course, I have told you in certain of plants, number of female flowers can be increased by the application of ethylene. Now, like in cucurbita. Okay, so number of increase in female flowers would mean number of increase in fruits, right? Last is sprouting of storage organs. In certain of plants like rhizomes, tubers, etc., these particular storage organs can be induced to germinate under the action of ethylene. Like potato tubers, they can be allowed to germinate under the action of ethylene. That can be beneficial to various agriculturists. Okay, so that being said regarding ethylene, next let us move on to the last phytohormone, that is abscisic acid. So abscisic acid, it is a mildly acidic form of phytohormone, and its function is completely a growth inhibitor. It is a growth inhibitor. Okay. Now the discovery of abscisic acid is also very interesting. Abscisic acid was discovered by 3 independent scientists, and they actually had discovered 3 substances having 3 different roles, but later on they found that all of these 3 that they had discovered were actually chemically and structurally similar. So that's when it was found that all the 3 can be given a common name, and it was abscisic acid. So let's see how it happened. So during mid-1960s, 3 independent researchers reported the purification and chemical characterization of 3 different plant inhibitors. One is inhibitor beta or B, that is of course a chemical that will inhibit various metabolic processes in the plant body. Abscission 2, of course it will be promoting abscission or shedding of the plant parts. And dormant, something that will be keeping the seeds dormant, seeds or buds dormant. Okay. So all these 3, even after they were found to have different functions, they were found to be chemically or structurally similar, and hence they were identified to be the same one single substance and were given a common name, that is abscisic acid. Alright. Now, occurrence of abscisic acid is dormant seeds and buds. They are, abscisic acid is mostly going to be confined to the dormant seeds and buds, hence not allowing them to germinate. And precursor is, it is a carotenoid derivative, I have already told you beforehand. So precursor to abscisic acid is viola xanthan. Okay. Now, moving to the functions of abscisic acid. Abscisic acid's functions would be, first is abscission. It promotes abscission of the plant parts like leaves, fruits, flowers. Dormancy of buds and seeds. By now I have told you so many times that it will be promoting the dormancy of seeds and buds. Metabolism. Now, what abscisic acid does is that it will inhibit the formation or synthesis of proteins or RNA. Hence, it will, inhibit the formation of chlorophyll. So, chlorophyll pigments would not be promoted, would not be formed under the action of abscisic acid. Of course, then there will be senescence happening, aging happenings in the leaves. Okay, so metabolism, mainly protein synthesis and RNA synthesis, will be inhibited under the action of abscisic acid, which will stop chlorophyll formation, leading to the senescence of leaves. Okay. Inhibition of seed germination. Of course, the seeds will be kept dormant under the application of abscisic acid. And closure of stomata. This is very, very important for your exam. See, it has been found that under stressful condition like water shortage in the plant or in the surrounding of the plant, abscisic acid's concentration would start increasing in the plant body. It is just like as if abscisic acid is the stress hormone to the plant. Whenever in stressful condition, abscisic acid's concentration will start to rise up in the plant body. So when abscisic acid's concentration increases in the plant body, automatically the stomata would become partially closed under the application of, or due to the abscisic acid being increased in the body, the stomata would start to close, not completely but partially. So as this happens, the water or transpiration rate will reduce, and that will kind of help the plant to survive water shortage conditions. Okay. So this is why we also called the abscisic acid to be stress hormone. Okay. Another very important thing is that because abscisic acid will inhibit the seed germination, and seed germination is promoted by whom? It is promoted by gibberellic acid, isn't it? Hence, abscisic acid is also known as anti-GA, against GA, so anti-GA. Okay, so do remember these names. It is also called stress hormone and it is also called anti-GA. Fine. Now moving to the usage. First is anti-transpirant. As you know, when you apply abscisic acid to a plant body, its stomata would partially close, and hence the transpiration rate will reduce. That will reduce the expense for irrigation. Okay, that will reduce the cost for irrigation. Hence, it is commercially important. Another is dormancy. You would agree that the basis to agriculture would be the seeds, isn't it? Without seeds, agriculture cannot be performed. And if suppose the seeds would have had this characteristics that they germinate immediately after formation, do you think you could have stored the seeds for growing in the next season? Of course not, right? So that's why it is very important that we can store the seeds, and that is only done when the seeds can be kept dormant. So by application of abscisic acid, we can keep the seeds dormant and store them for a long period of time so that we can use them for agricultural practices. Okay, so with that we come to an end here with the phytohormone part, which was the most important part. I hope all of you have understood it well. Now there is something that you should remember here. Now that you know the functions and uses of all the phytohormones, you would agree that certain phytohormones, they work together, like auxin promotes cell division as well as cytokinin promotes cell division. When two hormones are working in the same direction, they are promoting the same thing, we call such effect to be synergistic effect. Alright? Similar to that, sometimes you might have seen that certain phytohormones, they work against each other. Like take the example of auxin itself. Auxin is promoting apical dominance, but cytokinin is counteracting apical dominance, isn't it? So here again they are working against each other. That is called antagonistic effect. So after learning or after studying the entire phytohormone part, I think all of you are able to identify certain of important synergistic effects of phytohormones and antagonistic effects of phytohormones. I would suggest you to write as many as possible as you study the phytohormone. So as many as possible the synergistic effects and antagonistic effects by your own. If you are able to write them properly, your practice for this section will be done. Okay, so try to make a list of as many synergistic and antagonistic effects of phytohormone after you have studied it well. Okay, so with that, next class will be starting with seed germination and seed dormancy.